Hey everyone, so today we are going to talk about the synapsis of chromosomes in meiosis. Prompt 2. So here are some key definitions that we'll be discussing in the video: synapsis, crossing over, and paracentric inversion. Here is a live demonstration of the video that we are going to watch. Hey everyone, so this is a three-dimensional model of the first individual with its two chromosomes and this individual gametes that make up it. So as you can see, there are two extra regions on either side of these individuals, so you can tell that this is an inversion. This is actually a paracentric with an I inversion because the inversion happens outside or including the two chromosomes. So this is a paracentric inversion. This makes the recombinant products inviable, so that this means this would be a 50% of the genes would be inviable. If you do the inversion loop, you still can do an inversion loop with this, but it might look something like that. So the inversion loop can still happen. It would just look like this, and you would have to know that this is paracentric and that you would lose 50% of the genes because they would be inviable. Now if we look at this individual, individual 2, you can tell that this is the exact same, these are the exact same copies. They are just in different order. You could put these back and you would know that they still match up. They're the same copy. Therefore, all of these would be viable. Okay. So, the end result, we know that the individual 1 would have 50% non-viable gametes and 50% viable gametes. In individual 2, all gametes are viable due to the chromosomes being exact copies, and there's no aberration. So our conclusion, we can conclude that individual 2, the homozygous individual, will have more gametes that are able to survive and therefore will have the potential to produce more offspring compared to individual 1, which is heterozygous. Individual 1, we can conclude, has lower fitness than individual 2. Here are my references. Thanks so much.